Get ready. Stand up and run on the spot for 10 seconds. As you inhale, air travels through your lungs and deposits oxygen into your blood whilst carrying carbon dioxide out of your body. Congratulations, you are now participating in the phenomenon known as breathing. Now grab a straw and place it in your mouth. Block your nose and run on the spot for 10 seconds, this time only trying to breathe through the straw. Finding it difficult to breathe? Well, this is the harsh reality facing those who suffer from asthma. In its simplest form, asthma can be classified as a chronic inflammatory condition in which a person's airways become constricted. It's characterized by symptoms including shortness of breath, wheezing, chest tightness, and obstructed airways. However, asthma has become merely an umbrella diagnosis for a group of dysfunctional biological mechanisms that underlie a diverse group of symptoms, resulting in a disorder that is difficult to diagnose. The study of asthma has demonstrated how a single disorder can have varying distinct pathophysiological mechanisms. Any variance among these mechanisms can be classified into what is known as endotypes. These decipher the factors that contribute to asthma and what treatments can be implemented. This discovery has been made only recently and successfully explains why two individuals affected by asthma can exhibit similar symptoms yet respond differently to the same therapeutic treatment. This raises a big question. How are we supposed to treat asthma? Well, to answer this question, we need to quickly delve into the causes of asthma, everything everywhere, all at once. The star of this complex orchestra is the airway epithelium, otherwise known as the lining of the airways. The epithelium conducts an inflammatory response to either allergenic stimuli, such as dust or pollen, or non-allergenic stimuli, such as pollutants or pathogenic infections. As the action unfolds, specialized immune cells known as dendritic cells attack and engulf these stimuli through phagocytosis and present them to a naive T cell, calling upon his T helper 2 cell buddies to secrete chemical messengers known as interleukins, which fight off infections and rid the body of harmful pollutants. Unfortunately, during this process, a specific type of interleukin, known as interleukin 4, activates plasma B cells, which produce antigen-destroying proteins known as antibodies. In particular, a specific antibody known as immunoglobulin E, notorious for its role in triggering respiratory hyperinflammation, is produced in mass. To make this already complicated diagnosis even more convoluted, respiratory hyperinflammation is also associated with genetic mutations that induce airway remodeling, alter epithelial structure integrity, and allow allergic cluster formations. So, one very long and very convoluted story short, finding these associated mechanisms of asthma has not been easy. However, with the identification of a few key similarities, the overall asthma prognosis has been heavily influenced, leading to the development of vital treatments we now see in everyday life.
As you may have seen in movies, the media, or from a friend who has asthma themselves, we appear to already have a clear-cut treatment for the disorder. Ah yes, the one and only inhaled corticosteroids. The humble inhaler as many know it by, truly a marvel of scientific prowess captured in a small canister of multiple fun colors. The inhaler aims to reduce the impact of asthmatic symptoms on its user in two key ways. The first is through the strategic relaxation of small muscles within the epithelium, decreasing the sensitivity to stimuli in the long term. The second aims to reduce and/or prevent inflammation, providing rapid and immediate relief in the short term. Despite all of its benefits, inhaled corticosteroids also carry many inherent flaws. Although being fast-acting and effective for many individuals, they cannot provide relief for those with endotypes that are cortical resistant. Cortical resistance leaves those affected unable to control their symptoms, resorting to costly immunosuppressants, which makes them even more susceptible to pathogenic infections. Furthermore, prolonged use of inhaled corticosteroids proved problematic in its establishment of cortical dependence. Alongside this, the financial burden corticosteroids place on those without access to health insurance or just those within lower socioeconomic status is exacerbated. This financial burden individuals are faced with leads to underusing of treatments just to cover their daily expenses, making them vulnerable to their symptoms which they choose to persist through. Now you might realize these symptoms revolve around temporary reduction of symptoms, not addressing the roots. And with global prevalence, mortality, and economic burden deriving from asthma having increased significantly over the 40 years, do these treatments really represent the best solution? With such evident ineffectiveness in temporary treatments, are more further solutions needed? Well, let us now introduce to you gene therapy.
So, you're probably thinking, what even is gene therapy? No, not gene therapy, gene therapy. Gene therapy is the process of correcting dysfunctional genes correlated to a disorder by replacing or removing these specific genes to enable proper gene functionality. Gene therapy has become a modern remedial technique to manipulate biological activity by correcting dysfunctional proteins, removing mutations among DNA, with the aim to minimize the onset and impact of chronic diseases such as asthma. This closely aligns with BABS Theme 4, applying gene therapy to manipulate the function of genes through recombinant vectors and altering the expression of proteins within our bodies. This emerging medical technique holds great promise in developing permanent treatment of chronic diseases, providing remedy for symptoms unable to be relieved with current medical intervention. In time, potentially eliminating the worldwide incidence of disease, chronic illnesses becoming a thing of the past.
Now, it's fair to ask, how is it that inflammation of the airway, something that can be caused by random things from allergens to exercise to simply the weather, can be solved with a cellular therapeutic method like gene therapy? To consider this, we need to understand that inflammation itself is the body's own response to stress, as our lymphocytes, for example T cells, can activate cytokines, which are pro-inflammatory components of our adaptive immune system. This kind of stress can be a direct response to either an allergen, in atopic asthma, or more complex things like physiological stress or infection, non-atopic asthma. Asthmatics must have a component of their immune system that is not functioning as intended. A core culprit of this is dysregulated T helper cells that activate inflammatory cytokines, such as interleukin, specifically IL-4 and IL-13, which invade the airway, leading to asthma symptoms of airway inflammation. This means that if scientists could figure out how to go into our genome and block the signaling of asthma-causing allergens to our immune system, then the inflammation would be obstructed and the debilitating symptoms of asthma could theoretically be switched off. A study from 2003 confirmed that certain proteins could be created to block the activation of these cytokines, for example the IL-4RA protein, that could bind to the cytokines and deactivate them. This protein was delivered as gene therapy through a recombinant vector and administered through a mouse model. The results demonstrated a drastic reduction in eosinophil count, which are white blood cells that contribute to inflammation, conveying that this protein can effectively regulate the dysfunctional components of the immune system. This therapy can be used to greatly reduce some of the common symptoms of asthma, such as chest tightness, and overall can greatly improve the breathing quality for those that are suffering.
As decades of research continued, various studies began to realize treating the lung epithelium itself was the most effective treatment and reduced the risk of inhibiting systemic immune function for other areas of the body. By targeting the lungs directly using innovative methods to bypass the thick mucus barrier, untreated somatic cells of the body are left untouched. A study in 2020 demonstrated the efficiency of plasmid-encoded nanoparticles which contain an innovative anti-inflammatory hormone called thymulin. Thymulin can penetrate the mucus barrier of the lungs, directly lowering eosinophil concentrations and reducing airway inflammation in asthmatic mouse models, comparable to that of unaffected subjects. Involving only a simple direct inhalation, this innovation demonstrates an effective, efficient, and non-invasive modern gene therapy applications.
The reality is, asthma is such a common chronic disease as it has a very large amount of causes, and not all of them are genetic and can be fixed with the same methods, even with something as fancy and complex as gene therapy. While asthma that is diagnosed in childhood is typically due to these things, like dysfunctional T cells, DPP-10 proteins, or angry cytokines, other kinds of asthma can be caused by environmental factors. This could mean things like exposure to cigarette smoke, which is recorded to increase the level of IgE in the body and influence inflammation. Other causes can include weather changes such as cold and dry air, which for asthma sufferers can cause dryness of the airways and constriction, making breathing difficult and painful. These are just some of the branches of asthma that are still out of reach from gene therapy, showing that there is still a lot of asthma-related health risks that need to be considered. So, what can we make of all this? For decades now, there have been so many studies that present seemingly permanent, life-changing solutions, and our understanding of asthma and the immune system has expanded exponentially, meaning we should have all the answers, right? But there's one big problem, in that no present studies with these invasive gene therapy techniques have been made appropriate for human application, and there is still not a safe treatment that can switch off asthma symptoms for good. The long-term effects of these gene therapy techniques are still under scrutiny, as we cannot be certain if the long-term, unregulated expression of these modified genes will disrupt our normal immune function. While the future outlook of gene therapy asthma control is promising, we truly don't have the data that shows the safety of these revolutionary treatments, which means that for our current day and age, the safest and most effective gene therapy treatment to switch off asthma symptoms still eludes us.